Thank you everyone for joining our webinar today. Together with Sarah Batu, I have an honor to open the webinar for the International Day of Women in Radiopharmaceutical Sciences, which we organized under. And it's a second such webinar, so we believe it's becoming a tradition. So thank you, Sarah, for support in organizing it. And thank you to all the supporters. So a few words about PRISMAP, the European Medical Radionuclide Program, which aims at providing the access to radionuclide not yet broadly available. And women researchers play an important role in PRISMAP. So today it's my pleasure to introduce the first speaker, and this will be Hibak Mohamud, who is involved in PRISMAP research as well. So Hibak graduated in chemistry at the University of Surrey and did her PhD in radiochemistry in 2019. Currently, she's working as radiochemist on the development of medical radionuclides. Physical Laboratory in UK. So Hibak, the floor is yours.
Thank you. Hopefully I should be able to share my screen. Okay, I'll make a start. Hello, hi, I'm Hibak and I'm based at the National Physical Laboratory, and I'll hopefully give an overview today on some of the work that I do as a radiochemist here at NPL. So NPL is the UK's National Metrology Institute, and our main remit is to help with ensuring metrology is available to laboratories in the UK, and that's linked to labs worldwide. We're owned by the UK government, and we're funded by the Department for Science, Innovation and Technology. And the research that we do is across a broad range of fields. So for the group that I work in, which is the Nuclear Metrology Group, this was first established in 1913 to meet the demand for standardized radium. And over the years, currently now within radiopharmaceutical sciences, nuclear metrology is being used to help address issues related to reproducibility and validation in radiopharmaceutical R&D. And that is being done by us being responsible for establishing, maintaining, and disseminating to end users national standards. And this is all to support the growth in research in radiotheranostics. Hopefully this plot is probably one that a lot of people have seen already. But the search for new medical radionuclides continues, and access is limited due to challenges that are well associated with production, supply, and purity that are all being addressed within the PRISMAP project. So our role is to provide metrology, so that's traceable measurement to the becquerel. And there's a work package within PRISMAP, which is led by my colleague Sean Collins. That's all related to translating the data generated towards workable standards. So the example that I would like to give within this webinar are related to two that we've done within, which are available within the PRISMAP portfolio. So the first is actinium-225, which is used for targeted alpha therapy. And the second example relates to erbium-165, which is a promising radiolanthanide for use in theranostics. The key thing with each of these production routes is to keep in mind that during production that there are contaminants which get produced. So those are typically pseudo-isobaric interferences that are still present even after mass separation. But also you need to take into account the starting material, so the target that is irradiated to produce the medical radionuclide of interest. So prior to you doing any measurement or prior to you doing any work to produce your radiopharmaceutical, you need to ensure that there is a purification scheme in place. So this is the job that I have within MPL, and is the work that I do within PRISMAP. So a couple of facilities which help us to do this. The first one is obviously related to methods for purification. And to do that, typically you would use selective extraction chromatography resins. And generally what you would do is to work out a scheme which would allow for pre-concentration of your precursor of interest. And this is sometimes very challenging because the impurity or the target material is often much more concentrated than your isotope of interest. So an example of this is this elution profile, which I have here, which we developed from when we were looking at measurement of thulium-165 and the impurity gadolinium-149. And once you have this scheme in place, you're able to do method development with stable traces. And we're quite lucky at MPL to have a ICP-MS facility, which allows us to very quickly test these methods and to have these schemes in place. But we also are in a position to also be able to measure the long-lived impurity that is also present. And once we have a scheme in place, we validate this with the radionuclide precursor, which is supplied by the production facility. So hopefully in the next couple of slides, I'll give you the first example, which is related to the radiopharmaceutical erbium-165, which is supplied or is produced from the parent thulium-165. So why is purification needed? If you consider the pathway from your production to your final radiopharmaceutical, you can expect to have in the process some impurities. So if we take the example of erbium-165, so this is supplied to from CERN as a parent initially, so that's thulium-165, which is produced from doing proton spallation on a tantalum target. The thulium itself decays down to erbium-165, which is part of its decay progeny. But even after you do mass separation, you unfortunately still have a pseudo-isobaric impurity, gadolinium-149, which requires removal prior to you doing QC measurements and contaminant assay measurements. So at MPL, we developed a scheme which allowed us to have sequential elution of each of those isotopes. And once we were able to have this scheme in place, we were able to look at doing contaminant measurements of the impurity, so gadolinium-149, and we were able to reduce the breakthrough of this to unobservable amounts. So this was quite good because we're able to demonstrate a way of removing the impurity, and then you have your pre-concentration of your radionuclide precursor to then form your radiopharmaceutical. So this is why purification is really quite a key step. The second example relates to actinium-225, which is of growing interest due to its use for targeted alpha therapy, and again is one that is possible to be to get from PRISMAP. So this is done via high proton spallation on a thorium target, and over the past year we've received two targets of mass separated radium-225, which were then able to purify and get actinium-225. So my role again in here was to develop a scheme which allowed for high yields, so over 97%. The breakthrough of the parent was low, and we were able to have a reproducible method which allowed us to supply purified actinium to other PRISMAP labs that were interested in doing dosimetry studies or imaging. And the work within PRISMAP has also helped us within our own research program, where for the past year we've looked at production of actinium in the UK and taken the lessons learnt within PRISMAP to see if we can, with existing infrastructure in the UK, set up a way of producing actinium-225. So we have this current project that is ongoing with the University of Birmingham, where we have been able to get some experience with target preparation, and have done some initial studies looking at the use of the cyclotron facility over at Birmingham, where even though we know the amounts generated via protein spallation won't be comparable, we wanted to be able to assess the feasibility of production in the UK with this project. So using the existing methods we have developed, we've been able to do a similar work where we're able to demonstrate effective removal of radium-226 and have shown that it is possible to produce actinium-225, and hopefully with this is sort of the first step to demonstrate the need for further research in this area in the UK. So to conclude, I just wanted to say that radiopharmaceutical sciences is a very innovative field which has allowed for many opportunities for collaboration. The role of a metrology institute like MPL is to ensure that there are standards available to end users, and that there are quality control protocols which allow, prior to doing a standardization or a radio labeling, that there is a purification method in place that ensures that it is impurity free. So lastly, I just want to thank the many wonderful MPL colleagues that have helped over the years with PRISMAP, and the many good and helpful collaborators within PRISMAP as well. So thank you for listening.
Thank you very much, Ibak. Thank you. It's really a pleasure to have you as a first speaker. And as mentioned by Renata, I'm Sarah Batou, honored to be Deputy Institute Director of Nuclear Medical Applications at the Belgian Nuclear Research Center, and co-host with Renata and co-chair of this webinar. And you give Ibak a very nice way of introducing as a first talk some of the radiochemical methods for medical radionuclides, and to open the floor to this webinar. We'll have opportunity to ask you questions, but at the end of this webinar. Please note, everyone, that this session will be recorded and made available on PRISMAP platform. But thank you very much, Ibak. I would like to move now to the second speaker, another, I would say, young and junior scientist as a postdoc that we are very happy to have with us, Anna-Katrina Mapa-Nao, who is for the moment currently a postdoctoral fellow at the Center for Radiopharmaceutical Sciences at the Paul Scherrer Institute in Villingen in Switzerland. And before that, she did a postdoc on nanotechnology in Pisa in Italy, and was also a research intern at the Free University of Amsterdam in medical oncology in the Netherlands. So Anna-Katrina has got a very nice knowledge and expertise from different countries, different culture, and different subjects and areas that are related to radiopharmaceutical sciences. So I'm happy to give her the floor. Thank you.
Thank you very much. Just want to ask if everyone can see my screen, or is it okay?
Yes, but not full. Oh, yes, now perfect, perfect.
Okay. So thank you very much for the invitation and the introduction. As mentioned, I will be talking about terbium-149 for targeted alpha therapy in combination with somatostatin analogs. So, as many of us may already know, targeted radionuclide therapy makes use of agents that can specifically bind to overexpressed cancer biomarkers to deliver therapeutic radionuclides. This treatment modality is now an option for the care of patients with neuroendocrine neoplasms using the clinically approved radiopharmaceutical Lutathera that targets the somatostatin receptor. Like Lutathera, many other radiopharmaceuticals targeting this receptor and have reached the clinical settings are labeled with beta-minus emitters such as lutetium-177 and yttrium-90. Beta-minus particles have a tissue range that is in the millimeter scale and have low linear energy transfer, which is sufficient in treating large tumor masses, but not micrometastases and single cancer cells, which can be reasons for cancer progression. In addition, this rather long tissue range of beta-minus particles can deliver dose towards nearby healthy tissues. So there are a lot of alternatives proposed to overcome these limitations, and one of which is by using alpha-emitting radionuclides. And this is because alpha particles can deliver higher dose within their shorter tissue range that is in the micrometer scale. Alpha therapy targeting the somatostatin receptor has also been explored by others using the likes of actinium-225 and bismuth-213. Yet, when using these nuclides for therapy, one should critically consider their respective half-lives, as it is rather long for actinium-225 and short for bismuth-213, as this will have implications on the therapeutic outcome. In addition, the decay schemes, particularly that of actinium-225, involves alpha particle emitting daughter nuclides, and this can be of concern instead for the safety of the application. Instead, we at the Center for Radiopharmaceutical Sciences of PSI, in collaboration with Isolde in CERN, are highly interested in exploring the potential medical applications of terbium-149. This alpha-emitting radionuclide decays with a half-life of 4.12 hours, and importantly, terbium-149 decays into daughter nuclides with negligible emission of alpha particles. At this point, I would like to give you an overview on how we actually work with terbium-149. So PSI Isolde campaigns happen at most 2 times in a year, with each campaign lasting for 1 week. The work begins, of course, with the production of terbium-149 at the Isolde facility in CERN through high-energy proton-induced spallation of tantalum target and online mass separation. The product is then collected and transported from CERN to PSI, and these two institutions are roughly 270 kilometers apart, just to give you an idea as well on the logistics or importance of logistics. Upon arrival at PSI, radiochemical separation is performed to separate terbium-149 from the collection material and other impurities, and it is only after which that we can proceed with the in vitro and in vivo experiments that we have planned for the campaign. So as one can see from the end of the collection process at CERN to the beginning of the preclinical evaluations at PSI, we lose roughly 2 half-lives of terbium-149. That being said, the amount of activities used to perform subsequent studies must be adapted accordingly. Yet in the most recent campaign, which was last May, we were still able to accomplish a lot of these preclinical investigations, and of course this is thanks to the excellent quality and quantity of terbium-149 that we have obtained from our collaborators. We also would like to highlight here that this amount and quality of terbium-149 has greatly improved in the most recent campaigns. The use of terbium-149 for targeted alpha therapy has been previously demonstrated preclinically using a ligand targeting the prostate-specific membrane antigen for the treatment of prostate cancer and a conjugate that targets the folate receptor for the treatment of cervical cancer. In both studies, tumor-bearing animals injected with terbium-149 labeled compounds had delayed tumor growth, resulting in longer survival of these animals, as we can see from these survival curves. In addition to alpha particles, terbium-149 emits a substantial amount of beta plus particles, which can be taken advantage instead for imaging purposes. And now, knowing all this information, we sought to investigate the suitability of terbium-149 for the treatment of neuroendocrine neoplasms in the preclinical setting using somatostatin analogs known to have rapid pharmacokinetic properties. And for this, we use the receptor agonist dotatate, which is also known to be cell internalized, and we compare this with the receptor antagonist dota-LM3, which instead is known to be membrane localized. Experiments were performed in AR42J cells, which is a standard tumor cell model for investigations on the somatostatin receptor. Subcellular localization of these two radiopeptides were determined, and we confirmed that while both were highly taken up, internalization was only observed in samples incubated with terbium-149 dotatate. In the interest of time, I will be showing just the results from in vivo studies we performed using mice engrafted with AR42J tumors. And here we use the suitability of terbium-149 for PET imaging to determine tissue distribution at 2 hours post-injection. Both radiopeptides were found to be highly taken up in the tumors, and these signals that we see from the kidneys were attributed to excretion. Biodistribution studies were performed using the sister nuclide terbium-161, which is possible as these two radioisotopes have identical chemical properties. Again, here we see the high tumor accumulation of either radiopeptide accompanied by measurement of activity in the kidney samples, again because of excretion. Both radiopeptides were also found to be rapidly cleared from blood circulation. Therapy studies were performed in 2 parts. In study 1, mice were injected with 5 megabecquerels of either radiopeptide. This treatment resulted in delay in tumor growth, hence prolonging the survival of these animals. The median survival time for the animals injected with the radiopeptides, as indicated in the survival curves, is about 2 times longer when compared to the group of mice injected with the vehicle alone. Instead, in study 2, mice were injected twice on 2 consecutive days, with each injection amounting to 5 megabecquerels of either radiopeptide. So this intervention introduced further delay in tumor growth, hence prolonging further the survival of animals under repeated treatment. Yet, regardless whether applied once or twice, the differences between the effects of terbium-149 dotatate and terbium-149 LM3 were marginal. In summary, the short half-life of terbium-149 was found to be well-suited with the properties of dotatate and LM3. Subcellular localization was not indicative of the overall treatment outcome, as this was found to be comparable between the 2 radiopeptides. The therapeutic efficacy can be further improved by administering higher amounts of activity, but again, this will be depending on the amount of terbium-149 that is available. In addition, we can also take advantage of the repeated treatment or fractionated application of the radiopeptides, which clearly showed benefits based on the results on study 2. And finally, the effects of terbium-149 labeled peptides on healthy tissues and normal functions of other organs should be evaluated as well. All in all, the amount of studies exploring terbium-149 is still very much limited by its availability. Concrete actions must therefore be taken to enable further experimentations and potentially to bring terbium-149 closer for patient use. Such endeavor is envisioned by the IMPACT project proposed and planned by PSI, which aims to produce terbium-149 and many other medically relevant radionuclides. And with this, I would like to acknowledge everyone who is involved in this highly collaborative and exciting project. I would like to thank the organizers for the invitation and you for your attention.
The next presentation will be a video provided by our invited speaker, Mrs. Elena Mastantuono, who is the member of EU European Economic and Social Committee, and she is also a vice president of the section of Transport, Energy, Infrastructure and the Information Society. Recently, European Economic and Social Committee issued a nice letter information which also mentioned PRISMAP as a source of medical radionuclides, and we are happy to be recognized by you and also I hope you will enjoy the presentation that Elena kindly provided. So let's go for the presentation and then we will try again to connect Elizabeth.
Hello, my name is Elena Mastantuono. I'm the rapporteur of the European Economic Social Committee opinion on Europe's Beating Cancer Plan, driving forces for the security of medical radioisotopes supply. Did you know that every year more than 10 million patients in Europe benefit from nuclear medicine? However, for nuclear diagnosis and treatment, Europe relies heavily on third countries. These dependencies have the potential to disrupt supply chains and threaten access to life-saving medicine for millions of Europeans. The EESC has adopted an opinion outlining how Europe can beat cancer by beating the EU's strategic dependencies. Follow us through the preparation of the opinion. The work on the opinion started in the beginning of 2024 when the study group, consisting of 9 EESC members, met for the first time. Led by me and my co-rapporteur Philippe Chary, we firstly analyzed the basis for our considerations, Europe's Beating Cancer Plan, to get a deeper insight what problems medical centers are facing, but also what opportunities lie in nuclear medicine. We exchange views with experts from research institutes, industry and with patients' representatives, as well as with EU institutions. We have also visited several cutting-edge facilities, Geneva's University Hospital, the Belgian Nuclear Research Centre, SCKCN, and also CERN. The visits and feedback from the experts showed that the field of nuclear medicine is evolving rapidly, but satisfying the growing demand is challenging. Due to the short half-lives of medical radionuclides, it proves difficult to get them to patients in time, especially as the supply chain is also challenged by regulatory, technical, political, and financial obstacles. Furthermore, it was brought to our attention that access to the nuclear medicine is not equal for all patients in Europe, because of different systems of reimbursement, distance, socioeconomic conditions, and costs. We also enjoyed the exchange with the representative of the Belgian Council Presidency that picked up the security of supply of medical radionuclides, as well as reinforcing EU leadership in production as one of its priorities. Equipped with all the impressions from the visits, we gathered back in Brussels for the second study group hearing. We agreed that Europe needs better cooperation among member states, but also between the research centers, universities, hospitals, and industry in Europe in order to secure the supply chain of medical radioisotopes and allow for better access of patients to the nuclear medicine. We also highlighted the need to secure future supplies of high-assay, low-enriched uranium to build the needed production ecosystem in Europe, invest in the recycling and enrichment of source materials and stable isotopes, and to build a forecasting system for the supply of key medical radioisotopes. The next step in the life cycle of the opinion was to adopt it in the EESC section on transport, energy infrastructure, and the information society. Members positively assessed that the topic of medical radionuclides has been brought forward by the committee and suggested to follow up on that topic with the new EU institutions. Finally, the opinion was discussed and voted on at the EESC May plenary with 145 votes in favour and only 4 votes against. The opinion was adopted and sent to the EU decision-making bodies. On 17th of June, the Council adopted the conclusions that perfectly match with the recommendations given in our opinion. So what is needed to beat cancer and cut dependency for nuclear medicine? To save European lives, we need public and private investment in research and development and in new production infrastructures, sound regulation, and bold political decisions.
This is an important document that has been so supported by the European Economic and Social Committee, which is an important part of the EU. And most of the work has been conducted during the Belgian presidency that was taking place from the 1st of January this year to the end of June. So we are very happy that this is really an a very important achievement of this committee, but also very important for the community in terms of medical radioisotope supply and security of supply. Thank you very much to Alena Mastantuono and to all the committee. And the next speaker is Dr. Elisabeth Eppard, who is a radiochemist working on the development of process implementation of new compounds for theranostic application. And in 2013, Elisabeth obtained her PhD under Professor Roche at the Institute of Nuclear Chemistry in Johannes Gutenberg University, Mainz in Germany, where she worked on metal-based radiopharmaceuticals. After that, she joined Positron Pharma in Santiago, Chile, as a senior scientist in 2018. Then, since 2021, she has been a radiochemist in the Department of Nuclear Medicine, University Hospital Magdeburg, in Germany, where she's continuing her research. But she's also, and that's on that behalf that she will speak today, she's also a part of the EANM, Women's Empowerment Volunteers Pool, since 2021, and head of this Women Empowerment Internal Revision Unit since 2023. Thank you, Elisabeth, for joining us, and you have the floor.
Thank you again for the invitation to give a talk here, for your patience with my techniques, and thank you so much for the kind introduction. Yeah, today I'm here as representative of the EANM Women's Empowerment Initiative, where I'm representing the Internal Revision Unit. Actually, today we are celebrating with this webinar the International Women's Day in Radiopharmaceutical Sciences, and it's a worldwide celebration of all of us which are working in radiopharmaceutical sciences. So who are we? The Women's Empowerment of the EANM, or EANM-WE, initiative started 4 years ago with the first virtual meeting of the EANM-WE task force. And it was developed due to the great efforts from Professor Jolanta Konikowska as well as Professor Laura Evangelista. The WE is today an official EANM structure, which was also announced officially in April 21 as in the EANM channel as official structure. So the first team of the WE included several very, very passionate women about nuclear medicine, which are working in that field and want to enhance and improve our work life in nuclear medicine. So who was and is we today? We are a diverse group of women in age range, professional background, EANM involvement level, as well as scientific experience. So we have physicists, we have chemists, we have technologists, we have everything from young career researchers to high level senior researchers. And we have a variety of women active in the field which are willing to volunteer for the higher purpose of women in nuclear medicine. And we are in the very great situation that we are fully supported both by the EANM as well as the EANM Executive Office and our male colleagues. So why are we doing that? So actually, neither gender or professional or personal life choices should be decisive of the possibility to develop or take over relevant positions or participate in scientific activities, not in nuclear medicine or in science in general, but actually we are here in the field of nuclear medicine. So normally we are talking about EANM and the nuclear medicine sciences. Equality is key, not only in our field. So the current EANM, the Women's Empowerment structure, is pretty simple. So we have a leader, which is Professor Laura Evangelista, which is supported by two co-leaders, Agata Pieczak and Marika Kalnina from Poland and Latvia. And we have 4 units, which are supporting our leaders: the Continuing Medical Education Unit, the Internal Revision Unit, Professional Learning and Development, as well as social media. I guess if you're at LinkedIn, you will see or you have an experience of our social media unit, because we are posting a lot on LinkedIn and on other social media, just to raise the awareness about female figures in nuclear medicine and what's going on in the ENM with regard to women in the nuclear medicine field. So, for example, our LinkedIn channel, we have more than 300 posts related to this point up to now, and we are operating multiple activities and series of posts with regard to that point. We want to spread the knowledge about nuclear medicine procedures as well as in men's. We want to inform about activities, and we have a female figure series. So we want to raise the awareness about female figures, women, which impact nuclear medicine and science or medicine in general. And we are raising awareness for our volunteers pool. The volunteers pool exists since 22 and was developed and carried out by Lydia Kuna, which is now taken over by Marika Kalnina, and is one of our most important activities. We are constantly connected via WhatsApp group, and we have up to now more than 125 volunteer pool members. The volunteer pool is currently involved in all WE activities, gets invitations to the Congress, articles, revisions. We have a very strong data exchange. We have social media actions, as well as working groups, for example, for journal special issues. We can support personally. We have friendly chats, and if you need, get emotional support in the volunteer pool group. And this is important because actually we all know that there are inequalities which are in terms of access to personal, professional, or scientific development between women and men, also and especially in the field of nuclear medicine. And we want to overcome these inequalities. We want to promote the female face of nuclear medicine and improve the gender balance in the ENM's activities. We want to raise awareness for that and overcome them. And yeah, how do we obtain that? Actually, social media is nice, but it's not enough. So we are researchers, and we started surprisingly with research and publications. Up to now, 2 editorials as well as an original research article in Seminar Nuclear Medicine, all about gender balance, women in healthcare, women in cardiovascular imaging, and how to improve that. We had also a gender balance at workplace evaluation based on an international survey, and we have expectations towards activities of the ENM, and we evaluate these activities with regard to the impact. So the first editorial got over 300 replies from all over the world, and we had 14 questions with multiple answers possible to check, with the special focus on workplace situation, occupation, motivation, employment, and necessities in nuclear medicine. And actually we found out that we have a very heterogeneous group of considering the profession, age range, and residency, but all of them believe that women should be more represented within nuclear medicine. The most responders found it generally very hard or challenging to maintain professional family life balance, and 33% experience a lack of support from their co-workers or supervisors. 60% of our participants asked for training or help in training, focusing on self-improvement courses, support in researching skills and development, which is something which is together with normal research, not only true for nuclear medicine. And more than 50% of our respondents believe that more decisive actions shall be taken by the societies, not only the ENM, in terms of women involvement. So where is we going? We are started regular hosting sessions during the ENM annual congress. We are trying to extend our online ESMID webinars about women's topics. So already done is something about nuclear medicine and pregnancy, something about gynecological cancers, and we want to strengthen the connection on site the meetings with a volunteer pool and the heads of the We Women's Empowerment initiative. during the Congress. And actually, we are also, our volunteers pool is growing and growing and growing, and we are having now up over 125 members. We have more than 1,300 followers at the LinkedIn channel. We have achieved that more women attended the annual Congress. More women became E&M officers, and the gender balance within the E&M committees is improved too. And at the end, more chairs and heads of the sections at the E&M are women. How to reach us? It's very easy. So if you look for the E&M Women's Empowerment at LinkedIn, you can find us. You can send us emails over the office. You find us on social media, and we will always reply. Or you can directly contact our leaders, for example, Laura Evangelista. If you have any question or something, just contact us. We will answer you and try to answer every concern you have. And if you want to join the volunteers pool, you just have to send an email to the E&M Executive Office with the following information. Who are you? From where are you? What's your affiliation, your profession, your field of work and interest, and how did you find out about us? And at the end, we will respond directly to you and include you in the volunteers group and welcome them, because together we are stronger. Thank you for your
Thank you very much, Elizabeth. It was a great presentation, and I'm sure after this webinar, A&M Women Empowerment will get much more volunteers. So that's very encouraging. Thank you, and thank you for joining our webinar once again. And now I would like to introduce Dr. Aruna Korde from the IAEA. Aruna has a PhD in pharmaceutical sciences and over 25 years experience in the field. In her current role at the IAEA, she's responsible for coordinating research projects in radiopharmaceuticals, providing technical assistance to member states for capacity building in radioisotope production and radiopharmaceutical programs. And notably for this meeting, she is also a member of PRISMAP International Scientific Committee. And from the point of view of women in radiopharmaceutical sciences, she is coordinating this activity at the IAEA as an expert group under the Women in Nuclear Global. So Aruna, we are happy to host you today at this webinar. I hope you can switch on your presentation.
Yes, thank you, Renata, for nice introduction. And first of all, congratulations to PRISMAP for having this webinar, especially on women in radiopharmaceutical. And thank you for giving me this opportunity to talk today. See, as you all know, that radioactivity, because of its different characteristics, we have a lot of applications in different areas, and we at IAEA work in all these areas, and especially the health has the maximum applications. So amongst this, the nuclear medicine, I'm not going to go into details as all of we are into this area, but just to tell you that we work in all these aspects of radiopharmaceuticals, starting from the radioisotope production to the radiopharmaceutical development, preclinical, both in vitro, in vivo evaluation, and then process development, translational studies, regulatory, and for the clinical studies, our other colleagues in human health department, they take care of this, and we work in collaboration with them. So we have all a very much different activities in this area, and what all our mechanisms are like. We have the coordinated research projects. Currently ongoing are the 4 research projects, two in theranostic, two in therapeutics, and I think some of you are already the contributing members for the CRPs. Uh, then we support the technical cooperation projects for capacity building and human resource development. We have our different regular program activities with we conduct several conferences, meeting, and we work with WHO for preparation of the guidelines and regulatory things. We maintain the databases, develop the education programs for university syllabus, etc. So we come across, and all this we do with the help of experts and the member states researchers. So we what the gender equality or the empowering of the women is one of the sustainable goals of the UN, and our director general has he has introduced special measures for achievement of gender parity and especially for professionals in higher categories. So whenever we ask for the nominations, we encourage member states to identify suitable women participants for our meetings. We see that women participants get roles of chairmanship or the decision makers, and also in our in IAEA regular positions also. So, and as we know, the trends in radiopharmaceutical is the field is increasing. We just now also we heard the talks how the theranostic and the therapeutic uh, field is market is expected to increase, and there is also the venture capital investment in the area with the mergers and acquisitions of the big pharma coming into the in last few years. More role of the big pharma, and then the products coming from the like Novartis and other companies. So if you see the definitely there are many challenges, and one of them is like having the skilled or the educated and the capable workforce. So here recently the published articles on theranostics in Lancet Oncology. There is a survey that shows that number of radiochemists and radiopharmacists per million inhabitants from say around 189 countries. So here you can see that there is many of the areas in the globe shows much less availability of the trained radiochemist and radiopharmacist for to sustain these developments in the field. So there is a definitely there are more cyclotrons being installed at even at the universities and industry. Then there is a challenge to keep the academics and researchers going and to the radiopharmaceutical and radioisotope development area. As I will not be going into detail because just now you heard there are a lot of initiatives from the professional organizations like EANM, also the SNMMI, and Women in Nuclear Global is a non-profit organization which is working for supporting women in all nuclear sectors and nuclear applications. And there are few of the publications specifically addressing the role of women in nuclear medicine, in radiopharmaceutical. I think the one complete issue was dedicated to this. Then gender balance in nuclear sector. This report is recently published by OECD. And I would like to say that SNMMI, the latest, the current president is a nuclear medicine, is a radiochemist, and she's a very good supporter for our women in nuclear group. She's one of the advisors for this group. So why do we set up? What was the motive for setting up this network, because when I joined the agency, um, I realized that we have a lot of women in radiopharmaceutical, and we conducted the meeting, one big conference that is called ISTR, the Trains in Radiopharmaceutical, and we had more than 400 participants, and around 40 to 42% were the women. So we had around 200, around 150 to 200 women attending that conference. So during that, we thought of bringing them all together for one side event, and then we established this networking group. And then these women in nuclear global, we contacted them, and they allowed us to, or they were very happy to establish this as a first initiative group of, like, group of expertise in radiopharmaceutical. And now if you see, the Win Global has many different initiative groups where the women in particular area of nuclear sector are have come together, and they work together. So the main motive in this is to have the women in higher roles, even though there are many women working in radiopharmaceutical area, but the leadership role is not as many as expected. So we have currently the 172 members from 42 different member states. And when we analyze the questionnaire, what we have sent to them, you can see the interest or the overall experience of these women in different areas of radiopharmaceutical that apply to radiopharmaceutical. And we see that less than 30% members have said yes to experience with radioisotope production. And in this also, more of them have said experience in generator. So it may be just the handling of the molybdenum technetium generator, but actually the women working in the with the isotope production with the cyclotron or reactor, it's still less. And of course more number of is in the education or academia or hospital radiopharmacy. And so we there is still a lot of scope to work in many different areas and with the different collaborations. So in IST, in the next IST, we formed this group in 2018, 2019, 19 ISTR, and we had another conference after 4 years in 2023, where we had 48% women, and we had out of the 5 young investigator awards, 4 of them were the women achievers. So it was a very uh, a nice feeling to have the young women achieving the very good results in radiopharmaceutical. So we conducted the side event. The topic was like my voice and my message, which brought together the young and as well as the successful women scientists in the area. And similar way, we had the mentoring, speed mentoring event where we gave the opportunity for researchers. Of course, this was not only for the women, but we had more than 70% women participant into this. So it's like one-to-one interaction with the scientists and the young researchers. Recently, we conducted 2 webinars because of uh, the increase in the interest in radiopharmaceutical. There is always uh, things to know what are the opportunities in academics as well as what opportunities in industry. So we had these 2 webinars, and we brought academicians from different regions of the globe, as well as some successful women who are leading who have a leading roles in current radiopharma industry, from IBA, from Curium, from Novartis, and ITM. And we also had one women engineer who is as an independent consultant, we had the mentoring program from representative from Win Global, and for academic side, we had the young researchers and the senior scientists and professionals, as well as Win IAEA representative to talk about the initiatives of IAEA. And these are some of the messages. We have this recording of this webinar available on our website, and these are some of the messages given by these women, both from the academics and industry side, and these were very interesting ones to hear about their experiences, challenges, and what made them continue working and being successful. And before I close this, I would like to tell you about our very recent activity about the radiopharmacy database. And this is now available online. And what we need is we need the inputs from the all facilities, because this database will be updated based on the response to the survey which we have created and available online. So please go to this link and you can submit your, there are the instructions. So based on that, you submit the survey here, and this database is available for in 3 pages. One shows the radiopharmacy facility, another one shows the radioisotope facilities, and the 3rd page gives you all the information related to preclinical research area, what are the whether they have the health regulations available, what if there is the training facility available. So please kindly go to this if you are working on the radiopharmacy facility, and then fill the survey. And this database is for the worldwide facility. So this will be very useful to see the trends and what are the gaps and what are the challenges within the region or even within the particular countries. So the IAEA initiatives for all nuclear sector is the Marie Curie Fellowship Program. Currently it's open for application up to the 30th of September. So please visit this website. And there is also the Lisa Meitner program, but currently it is only limited to till last year. Definitely it was limited to the nuclear energy sector, but we hope that with more budget we may get a chance to have it extended to other fields as well. So we had the International Women Day, where all the Lisa Meitner and Marie Curie Fellowship awardees came together. And to tell you that we have only one radiopharmacist who is graduated to this IAEA Marie Curie Fellowship program, and it was so nice to see her, and she worked for 6 months with us as an intern. And this is what she says, that it makes a lot of difference to bring women together. So like this, things are very fruitful to have this net. Sorry. So to end that, I will say that whenever there are opportunities, say yes, and then you can figure out the rest. Thank you.
Thank you very much, Aruna. Thank you for this inspiring talk and on the role of women in radiopharmaceutical sciences and the crucial contributions women are making. And also it's very inspiring to see what EIA is doing to encourage more women to pursue their careers in this field. Due to the time constraints, it will be very difficult to handle the questions. So we would like to propose that the speakers answer writtenly to the questions that have been asked. And as we come to the close of this webinar, I would like first to thank all our distinguished speakers for sharing their expertise and their insight with us today. Would like also to thank everyone who attended and participated in this event. And we hope that all the presentations today were inspiring to you and provided you some valuable perspectives. And finally, a special thank to the PRISMAP team for their support in organizing this event. And also, last but not least, to my co-chair, partner in crime, Dr. Renata Mikowicak, for her wonderful partnership throughout the preparation and through this process of this Women International Day. So thank you once again, and we very much look forward to continuing this initiative and to meet you next year for the next International Women's Day in Radiopharmaceutical Sciences. Thank you.
Thank you.